In this video, we describe how to create a TMA and make a block using GalileoTMAtic, a fully automated tissue microarray. Tissue microarray, or TMA, combines 10 to hundreds of paraffin-embedded tissue specimens into a single paraffin block, allowing the simultaneous analysis of molecular targets at the DNA, mRNA, and protein levels under identical and standardized conditions on a single glass slide. TMA is an effective tool for high-throughput molecular analysis of tissues, facilitating the identification of new diagnostic and prognostic targets in cancer. It is also a tool in basic research, prognostic, oncology, and drug discovery. It provides maximal tissue preservation and use, considering probable limited and irreplaceable archival tissue samples. The TMA project starts by selecting a series of tissue blocks from archives which respond to research biological questions. This must be listed in an artificial file or LIMS, together with a minimum clinical data set for each tissue block. From the identified and airgram tissue blocks, a glass slide is made, stained with a Chini. The pathologist then can analyze the stained tissues layer using either a microscope or additional scanned image, or else run the HG slide through a digital scanner and allow the whole flight to be analyzed using a visual imaging software based on artificial intelligence. In this case, the artificial intelligence selects the area of interest that marks the core areas that should be extracted, and overlays the image of this layer with the image of the tissue on the tissue block. The marked image of each NI slides and respective extraction coordinates are imported in the GalileoTMAtic construction folder and used for the construction of the TMA block. At this point, having available all the donor blocks and corresponding tissue slides, we are ready to make a TMA block with the GalileoTMAtic fully automated tissue array. First of all, insert the recipient and donor cassettes into the 6-position holder, and then place the holder on the automatic stage. One of the new features of GalileoTMAtic is to be able to work with different types of tissue cassettes simultaneously, such as standard tissue cassettes for all types of tissues, macro used especially for prostate cancer. 96/384 microfuge plate for molecular analysis of tissues extracted near the respective core on the TMA block. This facilitates the comparison of the protein and molecular analysis of the cells from the same tissue area. The need of TMAtic checks if the punching needles are vented or broken. The needle integrity check is active every time the head mechanism rotates. This control is exclusive to the TMAtic platform and ensures that the end of the automatic Pangic cycle is not damaged by an abandoned needle or incomplete by a broken needle. After checking the integrity of the needles, the barcodes of all block orders into show cassettes are captured and displayed on the screen. This track is very important. It controls that all tissue blocks are part of the TMA project and part of the design. TMAtic starts with an automatic punching cycle. The first operation is to measure the height of all tissue blocks on the table, including the macro and microfuge plates. The first step is to clean the recipient needle and check the disamptive from residual paraffin. Position the recipient block in the correct position. Makes the hole in the recipient block. Moves the needle's mechanism. And cleanse the donor needle. Then the donut needle gets cleaned before extracting the tissue core from the donor block. If the style mechanisms detect that the tissue core is not in the middle, the tissue extraction is repeated, increasing the static compression force. This control is exclusive of the GalileoTMAtic, ensuring the all cores are extracted and placed into the recipient block. In case a microtretop plate is used as a recipient, Galileo will pick and place the tissue core into the selected tube and then claim the recipient. Then the automatic coding cycle will continue until all the tissue cores selected are extracted in place, including recipient blocks, which can be either the TMI block and/or a 96 or 384 microfuge plate. Thanks to the open characteristics of GalileoTMAtic. It is possible using a robotic arm to take the microfuge plate and place it into an automatic Rigwell ending system, Hamilton, from which several processes can be made, such as reagents aliquoting, DNA or RNA exception capping and decapping, tubes and plates centrifugation and NGS reaction preparation. The robotic arm can transfer the microtiter plate into an incubator or to additional image platforms like Oberata. As you can see, the GalileoTMAtic platform allows to make a TMA block, giving the opportunity to analyze hundreds of tissue cores simultaneously under the same conditions, and make a molecular and genetic analysis of each extracted tissue core from the same block and near the tissue core placed into the TMA block. Thank you for your attention. We hope that this video was informative on how to plan a TMA project, construct a TMA block, and how to analyze the spots. Please feel free to contact us for additional information or classic clarification.